Good day everyone. Today, I'll be discussing the urea cycle, its history, its biochemical reactions, how it connects with the TCA cycle, its regulation and energetics, and the clinical disorders associated with it. Since I'm a nursing student, I'll also connect this discussion to nursing practice, so we can see its direct relevance to patient care. The urea cycle, also called the ornithine cycle, is the main process by which our body removes toxic nitrogen in the form of ammonia. Ammonia is mainly produced from protein metabolism, and it's highly toxic to our cells, especially to the brain. So the body needs a way to get rid of it quickly. That's where the urea cycle comes in. It converts ammonia into urea, which is then carried in the blood to the kidneys and excreted in urine. As nurses, this is very important to understand, because in patients with liver failure, the liver cannot process ammonia properly, leading to a buildup in the blood. In patients with kidney failure, the urea may form normally but cannot be excreted. Both cases are dangerous, and we are the ones monitoring these patients for changes in mental status and interpreting their labs such as BUN and ammonia levels. The urea cycle was discovered in 1932 by Hans Krebs and Kurt Henseleit. Interestingly, this was the first metabolic cycle ever described, even before the Krebs or citric acid cycle that most of us know. This discovery was very important because it showed, for the first time, how the human body has a systematic way of detoxifying nitrogen waste. For us in nursing, this is more than just a historical detail, it explains why doctors order BUN or ammonia levels in the lab. These lab values go back to a discovery made almost a century ago. Now, let's go into the reactions. The urea cycle happens mainly in the liver, inside the hepatocytes, and it takes place partly in the mitochondria and partly in the cytoplasm. Step 1, ammonia, carbon dioxide, and ATP combine to form carbamoyl phosphate. This step is catalyzed by an enzyme called carbamoyl phosphate synthetase I, or CPSI. This is the first way nitrogen enters the cycle. Step 2, carbamoyl phosphate combines with a molecule called ornithine to form citrulline. Citrulline is then transported out of the mitochondria. Step 3, citrulline reacts with aspartate, another amino acid, to form argininosuccinate. This step adds the second nitrogen that will eventually leave the body. Step 4, argininosuccinate is split into two products, arginine and fumarate. Step 5, finally, arginine is split by the enzyme arginase into urea and ornithine. Urea goes to the kidneys to be excreted in urine, while ornithine goes back into the mitochondria to keep the cycle running. So for every turn of the urea cycle, one molecule of urea is produced, which carries away two nitrogen atoms. Now, why does this matter to nursing? If this cycle doesn't function properly, ammonia levels build up. This condition is called hyperammonemia, and it presents as confusion, irritability, drowsiness, and if untreated, coma. Nurses are often the first to detect these changes during monitoring. The urea cycle doesn't exist in isolation, it is linked to the TCA cycle, which is also known as the citric acid cycle. The connection is through fumarate, which is produced in the urea cycle and then enters the TCA cycle. Why is this connection important? Because it ties waste disposal to energy metabolism. When proteins are broken down for energy, ammonia increases, and the urea cycle and TCA cycle are both engaged. In the clinical setting, critically ill patients, like those with severe infections, trauma, or burns, break down a lot of protein. This increases ammonia production. If their urea cycle is overwhelmed or their liver is impaired, they may develop encephalopathy. Nurses caring for these patients must closely monitor their mental status and lab results. The urea cycle requires energy, about 4 ATP molecules per cycle. So it's energy expensive, but it's absolutely essential for survival. The cycle is regulated mainly in three ways. First, by substrate availability. If there's more ammonia, the cycle speeds up. Second, the CPSI enzyme requires a molecule called N-acetylglutamate to activate it. Without this, the cycle slows down. Third, diet affects the cycle. High protein intake increases activity, while low protein decreases it. From a nursing point of view, this means diet management is crucial for patients with urea cycle problems. For example, patients with liver disease often need a low-protein diet, because too much protein makes ammonia levels rise even higher. For patients receiving total parenteral nutrition or tube feeding, we must make sure protein levels are balanced to avoid overwhelming the urea cycle. Disorders of the urea cycle can be genetic or acquired. First, genetic disorders. These are rare but serious, caused by enzyme deficiencies in the cycle. For example, CPSI deficiency or ornithine transcarbamylase deficiency. Infants with these conditions may present with poor feeding, vomiting, lethargy, or even seizures. Nurses play an important role in recognizing these early warning signs and ensuring the baby gets prompt treatment. Second, liver disease. This is the most common cause of urea cycle problems. In cirrhosis or hepatitis, the liver cannot detoxify ammonia properly, leading to hyperammonemia and hepatic encephalopathy. Nurses here monitor neurological status, administer lactulose as prescribed, and teach patients to restrict dietary protein. Third, renal failure. In this case, the urea cycle may be working normally, but the kidneys cannot excrete urea. This leads to uremia, with symptoms like fatigue, loss of appetite, itching, and in severe cases, uremic frost. Nurses monitor BUN and creatinine, prepare patients for dialysis, and provide education on fluid and diet restrictions. Let's take a case. A 52-year-old man with liver cirrhosis comes in with confusion and disorientation. His labs show an elevated ammonia level. As nurses, what do? First, we assess his neurological status, looking for changes in consciousness or esterexis, which is a flapping tremor of the hands. Second, we administer lactulose, which helps remove ammonia through the stool. Third, we monitor the number of bowel movements, ideally 2 to 3 soft stools per day. Fourth, we educate the patient and family about following a low-protein diet to help reduce ammonia production. This shows how the urea cycle is not just biochemistry, it directly impacts our nursing care. To conclude, the urea cycle is the body's main pathway for detoxifying ammonia and maintaining nitrogen balance. It links with the TCA cycle, requires energy and regulation, and disorders in this cycle are common in liver and kidney patients. For us in nursing, understanding the urea cycle means being able to interpret labs like BUN and ammonia, recognize early signs of encephalopathy or uremia, provide appropriate interventions, and educate patients and their families. This cycle is not just chemistry, it's part of safe and effective nursing practice.